First, let's start looking at the tree of life. And how do we build a tree of life? And so we need to have some sort of nomenclature to understand this. And a tree of life is basically just a 3-taxon statement. Now, taxa are simply another word for a species that has a name and groups of species. So Homo sapiens is a name. It's a species and it's also a taxon. Mammalia, including all mammals, that's a taxon and it's a proper name. But we look at them in terms of 3-taxon statements and we ask, is A and B more closely related to each other than either of them are to C? And then we might ask, are A, B, and C more closely related to each other than they are to another thing out here called D? Now, these parts of the tree, right there and right there, we call clades, because this is a cladogram, and a clade refers to a segment of the tree of life in which it encompasses all of the descendants of this node right there. Now, we can derive these big phylogenetic kinds of questions and problems from the notion of speciation. So we can start with an interbreeding population here, where these little dots are males and the opens are, say, females, and they interbreed, but then their barrier arises, and these populations begin to become isolated, and in isolation, they begin to diverge. So now we need to discuss some more terminology, very important terminology. So we have the word clade, and a clade is a group of related species. And so on a tree, this is a clade, and it's called monophyletic. It came from a single origin. Over here, if we classified these groups here, this is a non-monophyletic group because it comes from different parts of the tree, from different clades of the tree. And so the goal of phylogenetic analysis is to discover these monophyletic groups. And this has been the history of tree of life research for decades now. And we do not want names on non-monophyletic groups because the information content is tied up into shared similarity with monophyletic groups. Now there's other tree language that will be very helpful for us. 2 taxa, 2 species, 2 mammal, 2 orders, 2 families that are each other's closest relative are called sister taxa. And there are nodes on internal branches. And then there's these long terminal branches and the tips of the tree of which we have our taxa. And here's the root of the tree right there. And so there's always going to be root. We use an outgroup when we're really interested in understanding the relationships of this in-group. Another aspect of phylogenetic trees is how to read them. So if we look at this tree right up here, F and G are each other's sister group. E is their sister group. D is the sister group to E, F, G. C is the sister group to D, E, F, G. B is their sister group, and A is the sister group to everything else. Okay, now we rotate some of these branches, and we go to this tree, and it looks real different. And to somebody that doesn't pay attention, they say, oh, the relationships are different. But then look very carefully, and you'll see F and G are each other's sister group. E is their sister group, and D is their sister group. So you have this clade right here, and C is their sister group, and then you have B on the outside and A down at the bottom. Now you can rotate them again, and you can come up to these relationships. And you can rotate them again and come up with these relationships. Yet they're all the same tree. They look different, but they're all the same tree. They signify the same phylogenetic relationships. Now there's another thing that's very important, and that is people have a tendency to think that we are the most exalted creatures on earth and we are at the top of the phylogenetic tree. But that's not a way of looking at trees. We can change these trees around. Here it looks like we go from the poor fish to a frog, to a lizard, to a mouse, and then we get up to the humans at the top of the tree. But there really is no top of the tree in many ways. So we do some rotation of those limbs, and we find out, well, humans are right in the middle. All right? So it's important not to use trees to say something is necessarily more primitive or more advanced than anything else. Now let's talk about how we're going to build trees. We have three hypotheses here for three taxa. That's all the hypotheses that you can have for three taxa. So you can have A and B are more closely related than they are to C, A and C more closely related than they are to B, and B and C more closely related to each other than either is to A. Now, hypotheses are tested by derived characters, and primitive characters cannot be used to test hypotheses. And on the next slide, I'll be showing you that. But we want to find characters here, here, or here that test these different hypotheses. And we want to look at in-groups and out-groups. So these three taxa are called an in-group, and we also want to go out and we want to look at an out-group too, to understand their character distribution relative to those. Now, here's the way it works. We create, we have a root of the tree because we're testing these three taxa, A, B, and C. And so we're looking for shared derived characters. And these black bars indicate shared derived characters. So character 3 brings A and B together, character 2 brings A, B, and C together, and character 1 brings all of them together because it's all shared. But now you can see that character 1, because it's in all of these, it's a primitive character. And we call shared primitive characters simplesiomorphies, shared derived characters synapomorphy. Just this jargon, but it's used all the time by evolutionary biologists. So we can see that character 1 is shared by all of them. So character 1 can't tell me if A and B are more closely related to each other than they are to C because they all share it. So primitive characters do not give you give you any information about what's related. And then finally, we'll also talk about homoplasy, which simply means that the character has arisen in multiple instances. And we'll have a good example of that on the next slide. So here's a kind of busy slide, but it tells you all you need to know in some ways about how we might go about building phylogenetic trees. And so we have a bunch of taxa, A, B, C, D, and we want to know how are they interrelated. And specifically, we're very much interested in the three taxon statement here. Is it DCB, DBC, and then A with an outgroup, BCD with A as an outgroup again. So the relationships of these three taxa, D, B, and C. So that's what we're very interested in right now. So we have a character matrix, and A has the primitive condition, unfilled primitive condition, and then there's the distribution of character states that we interpret to be derived. And they're scattered and then they're shared in different ways. So we can take the three different alternative cladograms, these are the three cladograms, and then we can plot these on these different trees. So for instance, here is character 1, and it's found in BCD. So we can put that right there. Well, it's found in this one. We can put that right there, and we can put it right there. Then character 2 is only found in taxon B, and that's an odd apomorphy. So it's going to be there, it's going to be there, and it's going to be there. But now, when we get to character 3, it's found in C and D. Well, we can put that on that tree, but C and D are not related to one another. So if D has that character and C has that character, on this tree, that character has evolved twice. And we can do the same thing for character 4. We can put character 4 on C on all of them because it's only found in character 4. And then we have another character, 5, that's found in C and D. And so we put, there's character 5 there, there's character 5 there, 5 there, and 5 there. And 6 is found in B and C. So this tree here has B and C together. So character 6 evolved once in that shared common ancestor, but evolved twice there and twice there. Now, if we count up the character changes, we see this tree only took 7 changes or steps on the tree. This one took 9, this one took 8. So this tree right here is more parsimonious, and that means it just took the fewest changes. And we want to choose in science those hypotheses that explain something with the fewest assumptions. That's what we have to understand, because as we add more characters, then our preferred tree could change. And that's the nature of science. You're constantly trying to test these characters. And we apply optimality criteria. So I was applying what is called minimizing the branch length. So minimizing the number of changes. And so I was using the minimum number of steps or changes on those trees under what we call that criterion of maximum parsimony, or just simple character parsimony. But there are other ways in which scientists now look at trees, mostly with respect to DNA distances. So there's all kinds of tree-based, model-based approaches that can be used to generate phylogenetic trees from, say, DNA sequences. So here's a tetrapod tree, and just to make it very simple, all tetrapods, from frogs to lizards, birds, and then mammals, all have 4 legs, hence called tetrapods. Before then, they didn't have 4 legs, but these have 4 legs, and therefore it's a synapomorphy, and they're grouped together. All of these organisms from here on up are called amniotes because of the way of the membranes that are in surrounding the eggs. And some of them have, mammals, of course, have mammary glands, they have hair, they also have single bone in the lower jaw compared to other of these tetrapods. So there are different characters we can place on here, and there are special biochemical characters that unite birds and the squamate reptiles, lizards and snakes. And then within mammals, there are carnassial teeth, which bring the carnivores together. The cheetah and the jaguar, for instance, together. Now, you can see from this tree, you can see it's highly predictive. So if a colleague of yours goes out and said, oh, I just discovered a new mammal. Well, from what you know about the phylogenetic relationships of mammals, you can make a prediction. Well, they're going to have mammary glands. They're going to have hair, and so forth. And this is really important for the discovery of new life forms, because if we start classifying those, then we can start predicting what are their properties. And that's exceedingly important. And we'll get into that in future lectures.